Perfect. Okay. So thank you so much to everyone that is joining today this presentation. So I'm going to talk about the last paper I published when I was doing my postdoc in Dr. Ruffel in Moffitt Cancer Center. At the moment, I did a transition, so I'm a research project manager in a different lab, but I'm still in Moffitt. So, but I'm really happy to be able to talk today about this work I really did during my postdoc. So it is going to focus in the inhibitory receptor TIM-3 and how it limits the activation of the cGAS-STING pathway and intra-tumoral disease by suppressing the DNA uptake. So I would like to start this presentation mentioning another paper that I published in 2018 in Cancer Cell about the role of TIM-3 in dendritic cells. So when we started this project, we wanted to see the different immune checkpoint blockades, as you can see here in the graph, CTLA, PD-1, TIM-3, TIM-4, BTLA, different of the checkpoint that they have been in immunotherapies in the last years. And we also wanted to see the expression of this marker for breast cancer in the different immune subsets. So we really were really interested about how TIM-3 and TIM-4, they were highly expressed in DC1s, but they were not that much expressed in other like T cells. There's a lot of research on the TIM-3 role on CD8, CD4 T cell, but was not that much that was done at the moment on dendritic cells. So we did a first trial when we were treating the cells with chemotherapy in combination with TIM-3 and TIM-4 and both of them. And we were able to see that both TIM-3 and TIM-4 together, they were having a role in the chemo treatment. But when we did TIM-4 alone, we didn't see this effect that much when we treat them with chemotherapy. However, TIM-3 alone was working pretty well. So that's why we started to focus mostly on TIM-3 and not TIM-4. So when we published this paper in 2018, we were having how TIM-3 is highly expressed in intratumoral disease, dendritic cell disease type 1, and also how this TIM-3 antibody was increasing the expression of CSCL9 in dendritic cells, and is going to have a potential role in the recruitment of CD8 T cells and in a better response to chemotherapy. At that point, we wanted to know what DC activation pathway is going to be the one that is regulated by TIM-3. So we start to have some research and reading in the literature, and we have this nice review from the Gajewski in 2017, when they were studying explaining the central role of dendritic cells and STING, orchestrating these anti-tumor T cell responses. So they start the first ideas of how perhaps the liberation of the DNA and other dams, they could be associating about the infiltration in dendritic cells and how it's activating the type 1 interferons and is releasing the CXCL9 and 10 expression. Also, we found these other publications in 2014 from the group of Wu et al. In immunity that they injected the B16CIY tumor cells in C57 mouse. There were no rejection because in all the wild-type mice, they was able to grow the cell lines. However, it was a highly increased, the cells that were more aggressive in the mouse that they were knocked out for STING and IR3. However, they didn't see this change in the TRIF that is more related to the toll-like receptor knockout. So that's when we started our history, and I'm going to start presenting the paper that we published this year. So the first thing we did was to do a time course about the CXCL9 expression by treating a splenic disease with a heat shock that was a supernatant that we created by killing, by heat shock the tumor cell lines. And we used this as a conditional media with or without TIM-3. And we were able to see that when we were adding the TIM-3 antibody, we were able to have a highly nice increase time dependent for CXCL9, mostly in disease type 1, but not in disease type 2. So the next thing that we did was to sort the cells. And when we have sorted for the DC1s and the DC2s, we did a nanostring analysis to try to identify pathways and also genes that they were related to the role of TIM-3. So we start to see that type 1 interferons was the genes like we can see here for interferon alpha and beta, CXCL9 and 10, but all the interferon signaling pathway type 1 was highly upregulated in the samples that they were treating with this heat shock and TIM-3 blockade. So next, we wanted to treat our cells with interferon alpha, beta, and gamma, and we were able to see that mostly in DC1, we can see a really also nice upregulation of CXCL9. So if we want to make this link between CXCL9 and we are assuming that type 1 interferon is related and mediating this, what we did was to treat our splenic dendritic cell with our supernatant of the tumor with TIM-3, but also add an inhibitor of the type 1, as we can see here in the graph down. And we were able to see that the CXCL9 expression was totally abrogated after the type 1 interferon inhibitor. So since some of the evidence were indicating that potentially the DNA could be involved in this activation of the type 1, the CXCL9, what we did was to treat our dendritic cells after the exposure to the heat shock supernatant with DNAs and RNases. And we were able to see that when we treated with DNases, we were not able to have the CXCL9 expression. So that started to make for us more sense that this response could be potentially highly mediated by the DNA that is being outside of the cells after the heat shock. Similar of this result we have been also doing, there is not the data here right now, but we have also do with other ways to killing the cell like irradiation, and we were able to observe similar results that the heat shock. So at this point, since STING is one of the molecules that is really, this pathway is really is the one that is recognized the double-strand DNA. So we wanted to activate STING in a different pathway. So we use a STING inhibitor, activator, sorry. So we use the 2,3-cGAMP and the 3,3-cGAMP, and we were able to see also the nice activation of CXCL9, but only in DC1s, not in DC2s. And the type 1 inhibitor was abrogating the CXCL9 expression. So at this point, what we did was to take splenic dendritic cells from different knockout mice. On the one hand, we have the STING and the cGAS knockout mice. And on the other hand, we were having all the different knockout that they are mostly associated with the toll-like receptor, that MYD88, TRIF, and MAPS. And we were able to see that all the mice that they were splenic disease coming from these toll-like receptor knockout mice, they were still secreting CXCL9 amounts in a similar way than the wild type. However, the STING knockout and the cGAS knockout, we have an abrogation of the CXCL9 expression. The only one that was missing here was potentially some of the complaints that we recently also have here is about toll-like receptor 4, because we know that can go between MYD88 and TRIF together. So if we are only doing the knockout for one, could be a reverse effect on the other. So it's the only one, toll-like receptor 4, that we didn't cover with these findings, but I think it's pretty solid about the STING and cGAS. So the only thing that I wanted to know is, it's important to have the knockout is coming from maybe the tumor cells. So at this point, what we did was to generate PYMTs, tumor cells, that they were knocking out for STING or cGAS and making a supernatant and killing and having all the DAMs and the DNA coming from these cell lines. And we were able to see that was not an effect. So because we still were able to stimulate the CXCL9. One of the other important things is we were doing a lot of work with the splenic dendritic cells, but we wanted to see what is the role on bone marrow-derived CD103 bone marrow dendritic cells. So we were not able to get the same good amount of CXCL9 compared to the splenic disease, but we still were able to get an increase in the CXCL9 expression in these bone marrow dendritic cells. Also, since we were having more of these bone marrow dendritic cells because the splenic, we were not able to get that many to be able to do a Western blot. So that's why one of the reasons that we moved to the bone marrow derived ones, we were able to measure by Western blot the phosphorylation of IR3, as you can see here. So we were able to see that when we are blocking TIM-3, we have a nice increase in the phosphorylation of IR3, pretty compared to the result that we can observe with an STING activator like DMXAA. So also we were able to measure by flow cytometry the expression of phospho-IR3, and we're also able to see an increase on that. So at this point, we were pretty solid about the role of the DNA and the role of STING in the pathways. So we wanted to check different in vivo models just to validate our findings. So the first that we did was to have a knockout for STING mice and also cGAS mice knockout. And we were treating always our chemotherapy with IgG and chemotherapy with anti-TIM-3 therapy. So we were able to see that the wild type mice, they were responding pretty similar to all the findings that we have before, but the knockout for STING and cGAS, we cannot see any effect of TIM-3 in the in vivo models. So on the right side, we have the kind of the more complicated in vivo that we did, that was trying to make a chimera where we were having injection of two different types of bone marrow. On the one hand, we have the XCR1 and wild type or XCR1 with a STING. So this XCR1 DTR, we are only able to deplete dendritic cells type 1 when we treat with diphtheria toxin. But since it's the first image, we still have some bone marrow that they are coming for the wild type. We still are able to see this effect. On the other hand, we have here down the one that we are treating with diphtheria toxin, the XCR1, and then the other part is a STING knockout. So we were not able to see any response. However, in the other figure, where we have a mix with the XCR1 with the STING, but we are not treating with diphtheria toxin, we still can see that the mouse that were responding to the TIM-3 therapy in combination with chemotherapy. We also try all the different toll-like receptor knockout models for MYD88, TRIP, and MAPS. And we were able to see that this knockout, they were not having major effects, and we were able to see the same responses to TIM-3 in combination with chemotherapy, since the tumor, they were also stopped growing in these knockdown mice. So we were showing all the time our CXCL9 expression, but we wanted to try to see and validate if we are able to also see the DNA and be able to track the DNA in the cells. So for that reason, what we did was to treat our tumor cells with EDU, so that way the cells, they are labeled, and we were able to do a click reaction to be able to detect the DNA of the tumor cells. So we validate by flow cytometry, as we can see here. We were able to see a nice increase of EDU DNA in the cells that they were treated with the TIM-3 therapy. And then we wanted to be sure if the DNA was going inside the dendritic cells or maybe was attached to the surface of the cell. So that's why we did two different treatments with DNAs at the very beginning or a DNAs after 90 minutes of incubation. So we see that if we treat with DNAs at the beginning, there is a total abrogation of DNA. We cannot see any DNA inside the cell. However, if we treat it just at the end, after 90 minutes of incubation, we can see still that we have DNA expression. So that means that the DNA was not on the surface, so was not able to be degraded because was inside the cell. We also had the ability to use this image stream technology in Moffitt Cancer Center. So we were able to label our DNA and also the different markers to check for dendritic cells. And we were able to see that the cells that they were treated with the anti-TIM-3 antibody, they have this expression of the EDU DNA, and we can really see really nice in this single cell, the expression of the DNA that we cannot see when we only treat the cells with heat shock and IgG. So here we have some of the confocal images that we were able to also take just to validate in a more solid way the data. So where we can see how at heat shock with IgG, we have minor of the DNA that we are able to detect. However, we can see there's really nice colocalization with the gap DH in the cytosol of the DNA in our dendritic cells. But at this point, we wanted to know about what could be the potential pathway that is making the DNA to enter in the cell. So we validate with different molecules. So here we have, for example, the ciliobravin-D, that is a DNA inhibitor, and is going to inhibit the phagocytosis. And we were not able to see any impact in the DNA uptake. However, in the bottom right part, we treated the cells with a dynamine inhibitor, that is the dinosaur. And we were able to see that it's a totally reduction of the DNA after this dinosaur treatment. So this is validating that endocytosis is playing an important role in here, and our DNA is entering through endocytosis into the cell. So between the different receptor that they could, the different ligands that they can be related to TIM-3, there is only one that is cross-related also with DNA, that is HNGB1. So we start to think that maybe HNGB1 could be the one that is binding to the DNA and then is going to make a competition with TIM-3, and is the one that they are making the DNA to enter into the cells. So we start to treat our cells also with HNGB1 inhibitor. And we were able to see that this HNGB1 inhibition was abrogating the CXCL9 expression that we were observing with our anti-TIM-3 inhibitor. So other than doing that way, we have been also working in a more synthetic way with beta DNA and with HNGB1 and TIM-3. And we were also able to see not in the same effect that we can see with the heat shock supernatant, but also we were able to see how this beta DNA was able to increase. So at this point, what we wanted to see is, okay, we can see a CXCL9 expression. So that's going to analyze the DNA uptake also after HNGB1 inhibition. So we were able to see that also the DNA was abrogating, and we were not able to detect the DNA in our dendritic cell after HNGB1 inhibition. So since these results were pretty promising, we also proceeded to do an in vivo study with an anti-HNGB1 inhibitor in combination with our normal TIM-3 therapy. And we were able to see that the HNGB1 inhibitor was reverting the effect that we were able to observe with TIM-3 in combination with chemotherapy. That started to make us to think that this potential role that HNGB1 is having is the one that is mediating this DNA uptake. So then we have this other nice experiment that we did in the lab. So in this experiment, what we did was to inject the tumors, let the tumors to growth, and then start to treat the mice with TIM-3 antibody. And then we injected in the tumor the heat shock labeled with our EDU in combination with HNGB1. So we were able to see that we were able to detect in the dendritic cells intra-tumoral the uptake of DNA, but if we pre-treated the heat shock with HNGB1, then we were not able to see the DNA anymore in the intratumoral dendritic cells. So on the other side, we have another experiment just to see the presence of the recombinant HNGB1, how enhance the activity of the beta DNA to bind to TIM-3 coated bits in vitro. So this suggested that the TIM-3 was negatively regulated the DNA uptake by competing with TIM-3. So since another of the ligand of TIM-3 is galectin-9, we wanted to see what happened if, other than using a TIM-3 inhibitor, we are using a Gal9 inhibitor. So we were able to see first in vivo that the results of both approaches, they were pretty the same. So galectin-9 inhibition was able to mimic the effect that we were observing with the TIM-3 inhibition. So for that reason, what we do was to analyze the CXCL9 expression after treating the cells also with Gal9, and we were also able to see this enhancement of Gal and CXCL9 pretty similar to what we observed with the TIM-3 inhibition. So one of the things that we did was to check the galectin-9 expression in splenic and tumor disease, and we were able to see that they have a really high expression of galectin-9. So it makes us to think that could be also a potential target. So what we did at that point was to use the CD103 bone marrow dendritic cell that they have a lower expression of galectin-9. So that way we were able to overexpress on these cells the galectin-9. So we were able to see that this recombinant Gal9 was not having an impact in the increase of the expression of TIM-3, but we can see a nice increase of the expression of galectin-9. So we also analyzed by confocal the effect of the galectin-9 overexpression, because one of the potential approaches that we were thinking why galectin-9 is having an important role here is because galectin-9 could have a role in making clusters of TIM-3, and that was the way that the cluster of TIM-3, they were able to compete easily with HNGB1 and not making able the TIM-3 to enter into the cells. So we were able to corroborate that findings by confocal. What we can see that the overexpression of galectin-9 is making this nice co-localization of clusterings of TIM-3 that we don't observe normally in the cells. So when we validated all the results with the mouse and the splenic for the mouse and dendritic cells for the bone marrow, we wanted to translate these results into humans. So that's why we start to repeat the similar analysis that we were doing, but isolating dendritic cells for healthy donors. We were able to see that repeating the same treatment with the heat shock, we were able to also have an increase in the CXCL9, 10, 11, and the type 1 interferon signaling. So we were able to check for the DNA uptake in different patient samples using the TIM-3 inhibitors, and we were able to see that the TIM-3 inhibition in human was also effective, since we were able to see an increase on the DNA uptake. Also, we measured the expression of CXCL9, 10, and 11, and we were able to see an upregulation and a high expression of these cytokines also in the human dendritic cells. So this is the last slide, just to summarize the findings that we published with this manuscript, how the TIM-3 blockade promote the endocytosis of the tumor DNA by the dendritic cells, how this DNA uptake and CXCL9 expression is each NGV1 dependent. Also, we show in this paper how the galectin-9 regulates the TIM-3 cell surface inhibitory function by making these clusters of TIM-3. And also the anti-tumor efficacy of TIM-3 antibody is closely related to the cGAS and STING pathway. So we can see here in this graphic how we have the normal dendritic cell on the left side on the left side. But when we are using these antibodies against TIM-3 and galectin-9, we are able to make the DNA to enter and not be sequestering in the surface of the cell. So being inside the cell, activating the cGAS and STING pathway that is activating this type 1 interferon, alpha and beta, increasing the expression of CXCL9 and 10, that they are highly related to the recruitment of CD8 T cells and enhancing the anti-tumor responses. I would like to say thank you for the Ruffel Lab. Dr. Brian Ruffel was an outstanding mentor, and I'm really happy I was able to join his lab when I moved here to the United States, and also all the colleagues I had in my former lab, Dr. Kai Hangi, Dr. Diana Celias, Alicia G. Olavisi, and Alexis. Also another researchers at Moffitt, Paulo Rodriguez, and the PhD students in his lab, Jimena and Islam, the Moffitt core for flow cytometry, microscopy, and the animal facility, and other Moffitt faculty at Vince Luca, Xiaoqin, and Hatton Soliman. And we have been closely related working with the Tesaro company, mostly for our human data. And this work was not be possible without also the financial and the help of the company. So thank you so much to all of you. And if you have any questions, that you can maybe type in the chat, and Eugenio will coordinate later the Q&A.
Thank you very much, Alvaro. And yes, if anyone has any questions, feel free to do it. I think I could also turn on your micro if you are really wanted to ask directly to Alvaro. So I will ask Alvaro my first question. So this is only on dendritic cells. Do you think this is also happening in other infiltrated immune cells in the tumor microenvironment? I don't know, like T cells, or I know also macrophages to have this expression of TIM-3. So you only show it on dendritic cells. Is this also happening in other immune cells?
So we mostly were focusing on the dendritic cell mostly because when